Greetings. I am Dr. Luis Isernia from the Universidad de Oriente from Republic of Venezuela. I have the honor of presenting my work entitled Effect of the Location of Active Acid Sites on the internal and external surface of different zeolites and active carbons based on petroleum delayed coke over the catalytic hydrolysis of starch. The scheme followed in this research consisted of analyzing the behavior of a series of zeolites with different ECAL ratios and different crystalline structures and comparing them with the behavior of several activated carbons obtained through different activating processes. These solids were analyzed using NHT-TPD technique to determine their acidic properties. Using methylene blue absorption, the monolayer capacity, absorbent equilibrium constant, and external surface area were determined. Additionally, catalytic tests were performed by monitoring the starch hydrolysis reaction and quantifying it using iodine spectrophotometry at 600 nanometers to determine the kinetic order, rate constant, and maximum conversion. For the kinetic monitoring of the catalytic tests of the starch hydrolysis reaction, a par 45 93 reactor with a capacity of 100 milliliters connected to a 4848 controller of the same brand was used. Masses of ore point or 164 grams of each catalyst were used at 150 Celsius degree, using initial starch concentration close to 1 times 10 to the power of minus 5 grams milliliters. The duration of each catalytic test was 12 hours, during which programmed a liquid of 5 milliliters were taken and analyzed by iodine spectrophotometry at 600 nanometers after adding 0.2 milliliters of KiI2 or 0.8%. In each 3 TPD experiments were performed in a laboratory built apparatus. Masses close to 0.1 grains of each catalyst were used. Initially activated at 873 K under a flow of 50 ml per minute of into at atmospheric pressure. Each activated sample was subjected to a saturation with NH3 at 50 ml per minute and 101 kilopascal of pressure for 15 minutes, and the excess was subsequently removed with a constant flow of N2 for 20 minutes. NH3 TPD experiments were carried out with N2 flows at 50 ml per minute and temperature increase of 10 K per minute until reaching 873 K. The ammonia dissolved in this step was trapped in a series of 15 milliliters vials with 0.1 normal H3BO3 were then titrated with 0.05 normal H2ECO4. The number of equivalent dissolved at each temperature provides information of the distribution of the active sites. The result of the catalytic studies demonstrate the greater activity of the derived carbons compared to zeolites studied in the starch hydrolysis reaction, as can be deduced from the graph of conversion versus reaction time. On the left side, which represents the behavior of the activated carbons, the conversion is seen to be practically unity, while in the case of the zeolites on the right side, the conversion reaches close to 0.6. Furthermore, in the attached table, the maximum value of the rate constants for activated carbons reaches 0.515 hour to the power of -1, while for the zeolites it reaches a maximum of 0.126 hours to the power of -1. Detail of the linearized graph of Ln1 minus x versus elapsed time, showing the higher k values derived from carbonate solids with respect to Zeolite series. Evaluating the effect of surface area shows a positive correlation between the external surface area and the rate, considering first-order kinetics for both the zeolites and activated carbons studied. This can be corroborated by observing the attached table, which clearly shows how increasing the external surface area leads to an increase in the K value. Likewise, acidity has a positive effect on the activity of activated carbons, as can be seen in both the graph shown and the attached table. Zeolites, on the other hand, exhibit undefined behavior, which contrasts with the behavior of activated carbons. However, the image 3 TPD profiles allowed to demonstrate the general trend in zeolites towards an increase in their activity with the increase in the strength of the acid sites, as shown in the attached graph. Preliminary results allow us to consider two distinctive forms of the interaction between zeolites and activated carbons with starch polymer molecules. In the case of zeolites, this distinctive behavior may be due to the proportion of acidic active sites between their micropores, which are almost inaccessible to the long polymer change of starch, making their conversion difficult. On the other hand, the results suggest the presence of a significant fraction of such sites on the surface of activated carbons, where starch polymer changes are directly observed with their glycosidic bonds arranged directly on the acidic active sites. In conclusion, in the Zeolite series studied, a higher proportion of active acid sites in their micropores, which are inaccessible to polymeric starch molecules, led to a lower catalytic activity than in the activated carbon series. In contrast, in the activated carbon series, the higher proportion of active acid sites on their external surface, which are more accessible, led to a greater catalytic activity. With this, we conclude the presentation. We hope you found it interesting and that we're happy to answer any question you may have. Thank you very much.